Hello everyone, this is Dr. Prashanth and welcome back to Public Health Series. In this video, I'll be talking about vaccination. Vaccination is a simple, safe and effective way of protecting against harmful diseases before we come in contact with them. It uses our body's natural defenses to build resistance to specific infections and makes our immune system stronger. Coming to the mechanism of action, initially it recognizes the invading germs such as virus or bacteria, then produce the antibodies. Antibodies are nothing but proteins produced naturally by the immune system to fight disease. And lastly, it remembers the disease and it starts fighting. If you are then exposed to the germ in the future, your immune system can quickly destroy it before you become unwell. The ingredients of a vaccine include antigen, which is a kill or weakened form of a virus or bacteria, which trains our bodies to recognize and fight the disease if we encounter it in the future. Adjuvants help to boost our immune response. They help vaccines to work better. Preservatives ensure a vaccine stays effective, and stabilizers protect the vaccine during storage and transportation. Now coming to the type of vaccines, first is the live attenuated vaccine where the pathogens like virus or bacteria are weakened by genetic manipulations to limit its growth and thus do not cause disease to the host. The weakened pathogen generates a broad immune response in the host like that shown by an infected individual with a natural pathogen. Examples include oral Sabin polio vaccine, measles, mumps, rubella and varicella, nasal influenza vaccine, BCG and rotavirus. Next is inactivated vaccine or dead vaccine, where the disease causing pathogen is killed or inactivated, usually through a thermal or chemical process. Such vaccines, when administered, elicit a robust immune response that mimics most of the responses seen during an infection. Examples include typhoid vaccine, influenza vaccine, Salk polio vaccine, hepatitis A, and whole cell pertussis. Next is acellular or subunit vaccine. Acellular vaccines do not contain the whole bacteria or viruses. Instead, they contain polysaccharides or proteins from the surface of the bacteria or virus. These polysaccharides or proteins are the parts that our immune system recognizes as foreign and evoke immune response against them. Examples include hepatitis B, acellular pertussis. Coming to conjugate vaccine, like subunit vaccines, conjugate vaccines use only portions of the bacteria or virus. So basically, bacterial polysaccharide molecules are chemically linked or conjugated to a carrier protein. Examples include Haemophilus influenza type B, pneumococcal conjugate vaccine, meningococcal conjugate vaccine. Next is toxoid vaccine. Some pathogenic bacteria release toxins or poisonous proteins when they attack the body. So some vaccines are made by inactivating these toxins chemically and called toxoids because they look like toxins but are not poisonous. Examples include diphtheria vaccine and tetanus vaccine. Now coming to DNA or RNA vaccines, genetic material, either DNA or RNA, from the pathogenic bacteria or virus is introduced into the human cells and the cell machinery is employed to produce the protein encoded by the inserted gene or genes of the pathogen. Our body's immune system detects such protein as a foreign agent and produces an immune response against the whole pathogen. Examples include HIV vaccine. Next is recombinant vector vaccines, where a small piece of DNA is taken from the disease-causing bacterium or virus, and this gene is incorporated into a plasmid or a carrier vehicle, which enables production of large quantities of well-defined proteins, which are then used as vaccines. Examples include HIV, rabies, and measles. Now coming to mRNA vaccine, it is one of the newest and most exciting areas in the vaccine technology. When an mRNA vaccine is delivered, the RNA material teaches our body how to make a specific type of protein, but does not make the person sick. The protein triggers an immune response, that is through generation of antibodies, and in that way, if a person is ever exposed to that virus in the future, the body will like to have the tools like antibodies to fight against it. Examples include COVID-19 vaccine. Next is viral vector vaccines. Viral vector vaccines use a harmless virus to deliver to the host cells the genetic code of the antigen you want the immune system to fight. In doing so, information about the antigen is delivered, which triggers the body immune response. Examples include Ebola vaccine and COVID-19 vaccine. And now coming to the history of vaccination, in 1700s, the smallpox vaccine was discovered, and the important discoveries in 1800 are cholera vaccine, rabies, tetanus, typhoid fever, and bubonic plague. And the important discoveries in 1900 are pertussis, TB, diphtheria, yellow fever, influenza, BCG, polio Salk type, Japanese encephalitis, and the polio Sabin type in 1962, followed by measles, mumps and rubella, and the expanded program of immunization was launched in India in 1978, followed by universal immunization program that was launched in 1985. And in 1999, the first vaccine for rotavirus was discovered. And now coming to important discoveries in 2000, HPV vaccine, that is in 2006, hepatitis E 2012, malaria in 2015, dengue fever 2015, Ebola 2019, and COVID-19 that was in 2020, and the recent discovery was chikungunya that was in 2023. Now let's look at the National Immunization Schedule for infants, children and pregnant women. To start with the pregnant women, three vaccines are given: TD1, TD2 and TD booster, tetanus and adult diphtheria. The first dose is given early in pregnancy, followed by TD2 4 weeks after TD1, and TD booster is given if they receive 2 TT or TD doses in pregnancy within the last 3 years. All the three vaccines are given intramuscularly to the upper arm and the dosage is 0.5 ml. And for infants, BCG, hepatitis B, and OPV0 are given at birth, and OPV1, 2, 3 are given at 6 weeks, 10 weeks, and 14 weeks, followed by pentavalent 1, 2, 3 at 6 weeks, 10 weeks, and 14 weeks. Next is pneumococcal conjugate vaccine, two primary doses at 6 and 14 weeks, followed by booster dose at 9 to 12 months. Next is rotavirus at 6 weeks, 10 weeks, and 14 weeks. Next is inactivated polio vaccine, IPV, two fractional dose at 6 and 14 weeks of age. Next is measles rubella, that is MR first dose, given at 9 completed months to 12 months. Next is Japanese encephalitis JE1, given at 9 completed months to 12 months, followed by vitamin A first dose, that is given at 9 completed months with measles rubella. And now coming to children, diphtheria, pertussis and tetanus, that is DPT booster 1, and MR, measles rubella second dose, OPV booster and Japanese encephalitis second dose. All these 4 vaccines are given at 16 to 24 months. Followed by vitamin A, second to 9th dose, that is given at 16 to 18 months. Next is DPT booster 2, given at 5 to 6 years. And TD, that is given at 10 years and 16 years. Okay, this is a simplified version of the National Immunization Schedule. It just includes the vaccine and the time it is given. So you can just go through it. Thank you.